without delaying more. Okay, so I welcome you all, sir and madam, in this science dialogue series. As you all know, this is free webinar series. We invite experts from different domains of bioscience subject. Main purpose is to increase knowledge pool and increase networking also, if we are capable to utilize this network. So in this series, we have invited Dr. Rahul Verma. He is recently assistant professor at Amity University, Bangalore campus. And before joining here, he did his postdoc from Florida, PhD from IIT Indore, and MSc from Jawaharlal Nehru University. So he has very good credential, academic background. With this brief introduction, I invite Dr. Rahul Verma in this Science Dialogue series to deliver lecture, and I am grateful to you for accepting our invitation and giving your sharing your knowledge and time. I request participants to attend complete lecture and fill feedback form also. We will share that Google feedback form in this chat box, not after, not before the lecture. So if you need e-certificate, then fill the feedback. Otherwise, no need to fill the feedback. Keep in touch through social sites. We have different social platform: LinkedIn, YouTube, WhatsApp group, Facebook. Uh, there are blogs. So you keep in touch so that you will not miss relevant lecture. Most of the lectures are free. Sometimes we organize short courses. Like recently, our one course is on biostatistics, second course is on bioinformatics. So you can also attend that. And there is guarantee of the quality of the content. So with this brief introduction, why people are joining and leaving, I don't know. So now, Dr. Rahul Verma ji, now platform is yours. I welcome you in this Science Dialogue series. Are you listening? Are you here? Dr. Rahul Verma?
Yes, sir. Yes,
Yes,
I am
please.
here.
Yes, please start.
Thank you, sir, for inviting me for this wonderful science dialogue series, and gave me the opportunity to talk about complex networks and biological networks. It's a very new field in the domain of biology. It's a very old subject in mathematics and have been extensively used in physics also. But in biological systems, because of availability of huge data now, we are able to apply the principles of network science in biological systems. And as we all know, the biological systems are very complex and have uh, huge data to work with. So because of this principles of network science, we are able to get more and more information from the interactions between biological components, and those interaction gives some uh, results, and from those we get more information and look at the overall picture of the cell, because in a cell not everything is working individually,
Full
all the
slide mode
things.
होगा.
Yes, sir.
जी 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 कर दीजिए इसको.
Yes, sir.
Yes,
So,
yes, very good.
in biological systems, no component is working by alone, by individually. Everything is interacting with everything. So, for as a network scientist, as a network biologist, I see the whole cell as one complex system comprising of many different networks. So, I will Today, in this Science Dialogue series, I am grateful for this opportunity, and I would like to talk about networks in general, and how biological networks are constructed, created, and what are the different properties that we study. And at the end, I will talk a little bit about my work that I have done using mitochondrial genome and network science. So, with this, I would like to start my presentation. Okay, so first of all, as when we say network science, usually people think of computer and other things, but network science is actually a field of physics that stems out of graph theory. So graph theory was is a very old concept in mathematics, and this problem came because of a simple question. In Königsberg, there was a place called Königsberg. So in that place, there was a river called Prigel River. And on that Prigel River, as you can see in this picture, there are seven different bridges are there. So there were some people, intellectuals, they asked, can we go like through the city and crossing each bridge only once? So this was like a very simple communication, simple talk, but that gave rise to a beautiful concept of graph theory that we study. So Leonard Euler, I think you have heard of the Euler scientist. He was very renowned person in mathematics. So Leonard Euler gave a negative solution that this is not possible. We cannot go to each part of the city crossing the bridge only once. So we have to go to one bridge at least twice. So this gave a foundation for graph theory. Now, why it is like this? So, by simplification of this picture, you can see here like they simplified it, and then later on, this is converted into as a graph, as you can see here. So these links are these green dots are land that you can see here. This is small part, this and this. So there are four land masses, like one here, one in between here, one here, and the other one is here. So four land masses are there, which are connected by seven bridges. So you can count 1, 2, 3, 4, 5, 6, and seven
आप
bridges are
उसमें
there.
प्रिंटर का ये पॉइंटर का ऑप्शन है वहां नीचे जिसमें वो लेजर, हाँ हाँ वो मोटा थोड़ा सा ले लीजिए तक नहीं, इधर इधर पॉइंटर का, हाँ हाँ थोड़ा ठीक है, यस।
Okay, sir. So yes, so you can see that there are seven bridges. So because it is odd number, now you see in this there are four. These green dots are there. I will tell you what we call this, and these bridges are there. Seven bridges are there. So because of this odd number, we cannot go to each this green dot without going to one of the links twice. So we have to go it twice. So this gave rise to a simple phenomena, and this was later founded as a graph theory, which is studied in physics as network science, and then later on it has been applied in biology since 1990s. There was very beautiful paper published by Albert Barabasi, and he studied protein-protein interaction networks in that particular paper, and that gave opened beautiful aspect of biological interaction network, interaction, protein-protein interaction as networks. And now it is a very flourishing field. People are applying machine learning, people are applying quantum biology with network biology and coming up with new ideas. So this is like a very booming field of physics which is applied in biology. Okay, so what is a network? Like we discussed about what happened and all that, but still, what is a network? So mathematically, since it is coming from mathematics, we have to understand mathematical concept of it. So mathematically, a network is a set of nodes and edges. So set means no node is repeated, no edge is repeated. 1, 2, 3, 4, 5, 6 nodes are there, and there are some edges. It's a simple set. Now, for a biologist or biology student, this is like very dry. to understand. But in our upcoming slides, I will explain how we look at the network through the lens of biology. So this network is a set, and it is represented by an adjacency matrix. Now, matrix, as you can see here, it is a simple representation of values in rows and columns. And adjacency means we are talking about two adjacent individuals. Suppose 1 and 2 are there, and 1 and 2 are connected. So 1, 2, 3, 4, 5 components are there, as you can see here. 1, 2, 3, 4, and 5 are there. Similarly, on vertical in the column also we have 1, 2, 3, and 4, 5. So interaction between 1 and 1 is seen here. So, and 1 and 2 is seen here. 1 and 3 is seen here. So you can see 1 and 2 are connected, so we put 1 here. And 1 and 3 are connected, so we put 1 here. But 1 and 4 are not connected, so it is 0. Similarly, 1 and 5 is not connected, so it is 0. Now, usually this matrix contains zeros and ones. So 0 means it is not connected, 1 means it is connected. And hence, this kind of matrix is called as binary matrix. It has only two values, either 0 and 1. So, if I and J are two individuals, if they are friends together, we put 1 in this matrix. If they are not friends, we put 0. Now, when we started working on network science, initially people have worked on social networks or clubs. Like social media came today, but earlier there were clubs. So who all people are going to club? In that club, who are friends with whom? So like that, they started working on social groups, and later on it become came to digital social media and digital platforms. So mathematically, we have understood, okay, this is a network, this is a matrix. But still, what is a network? We are not very sure so far. So when we look at a system, so system is, suppose a system is a population. Okay, there are some people in a city. Suppose Delhi, there is a city, people are there, and we will see, okay, there are so many individuals. Any two, if they are interacting, okay, so individuals are nodes here, and if they are interacting or they are friends, we put a link between them, or it is called as edge. So any two or more entities of any system, suppose we talk about cell, in the cell there are many different networks, we can say. So to keep the network homogeneous, we take only one type of biomolecule. For example, we are taking proteins. So that network will consist of only proteins. So it is a homogeneous network. Now, if two proteins are interacting physically or somehow because of some chemical reaction that they are performing, then uh, they, because of this interaction, they are performing some specific function. It is not that just they are just interacting and not doing anything. Then construction of network should have a purpose, that why we are constructing it. For example, proteins interact in signaling, cell signaling. So we will talk about signaling in that sense. Or if they are interacting for some metabolic process, then we will talk about metabolic reactions there. It is not like we are just looking at the interaction without any purpose. So they should perform a certain well-defined function. Then only this kind of interactions constitutes a network. So as we know, humans consist of approximately 20 to 23,000 proteins are there. Not all the proteins are present at one place, or not all the proteins are interacting all together. But there are some proteins interacting together to perform a function, and then we can make a network out of it. So these kind of networks we can make. Now this is a very easy and linear way of making a network. Like two proteins are interacting, we are forming a network. Now suppose there is a list of proteins we know, but we don't know whether they are interacting or not, because identifying the interaction between proteins is done through experiment only. Now we have a large database that, okay, these many proteins are there, they are interacting. Now from this information, can we find that whether two proteins which we have not tested whether they are interacting or not. Can we find that whether they are interacting or not? So these kind of questions are also asked that I will talk later in this presentation. Cell, there are not only proteins, not only biological components, there is DNA. There are other things also. When we work on it through mathematically or through other things, we can make networks. Like in my last slides, I will show you how I have made networks, not using a particular component, biological component or that, but using some numbers that is relating to biological system. So very initially, first biological network which was studied was protein-protein interaction network, and in that nodes are proteins. So nodes are the green dots that I showed earlier, and the edges are the bridges, which are either physical or functional interaction that we discussed right now. So this kind of network represent how the proteins are interacting. Now you will say, sir, we already know that this P53 is interacting with HIF1 alpha, for example, okay, or some other gene regulatory protein. But you are looking at only two or three proteins. Here, what we are trying to do is look at the bigger picture, because that's what I initially started. No protein is working by itself, it is working through a network. Like one information interaction is happening, and that information is being passed to the next level. There some interaction is happening. So this is how it works. So networks gives us a bigger picture. So we are going from top to bottom, not from bottom to top, like that. And if you want to go and look at the protein-protein interaction, you can go to string database and biogrid database. These are like commonly referred databases for identifying the protein-protein interactions. Next are gene regulatory networks. Now, gene regulatory networks have two components. One are genes and the other are transcription factors. Now, in this kind of network, this is not homogeneous. These transcription factors are basically proteins, and genes are, as we know, is DNA. So, this is what transcription factor is regulating what gene. Either it is activating or it is inhibiting. So, what we are looking here is the regulatory relationship through activation or through inhibition. So these kind of network are heterogeneous in nature because they have two different types of biomolecules, DNA and protein in it. And also they have two kind of interaction, either positive interaction or negative interaction. So this is adding more complexity to the network itself, because here we get a very simple network. There are only proteins, simple interaction is there, no positive negative, nothing is there. Here we have two type of biomolecules interacting, and second we have positive and negative interaction. So in this case, some transcription factors are being controlled by other proteins. So this will have connection with genes also, and this will have connection with other proteins as well. So this is a kind of more complex information present, and it is giving more information. So this, in this kind of network, we are interested in looking at the regulation of the transcription factors as a whole. Like individually, we can talk about few genes and few transcription factors. Usually, we talk about only one or two genes which are related to some disease or some particular function. But here, when we are making a network, we are looking at a bigger picture, that how gene regulation is happening in a particular state of a cell. Suppose cell is in or some organism is there in larva stage. We collect some cells and do the this analysis and find, okay, gene regulatory network make. So we make a network. We see, okay, this kind of pattern is there in this network, but at another mature stage or adult stage, we make another network and we compare. So basically, we are identifying the pattern. So for this kind of network, you can go to Transfec and Reg network databases. These are like see, the big problem with biological systems is collection and curation of data. We have to be very sure that okay, this is happening, and then only it is updated in these databases. So far, string and biogrid are highly updated, recently updated databases, but others are people are working on them. Next step, we have metabolic networks. So here, like as you can see, metabolic means there will be metabolites, which are some chemicals and enzymes interacting together. And here we will talk about the metabolic reactions, for example, glycolysis, Krebs cycle, and other such kind of chemical reactions involving multiple enzymes, reactants, and substrates and the products. So this kind of network will be studied for um, the interaction or biochemical reactions performed by metabolites. So this is important when we are discussing about dynamics of cellular compounds. Now, what is the dynamics? See, a cell is like a big railway station. Okay, people are going, people are coming, train is coming. So much things are going on all together. So it is a very dynamic, although it is in equilibrium, but it is dynamic. Whatever is coming in, some things are going out, things are coming in, things are going out. So this is dynamic, but it is in equilibrium. So to maintain that equilibrium or homeostasis, we study the metabolic networks. Okay, in some disease, some metabolic network will change their pattern, and we can say, okay, this is happening under this disease or under this state. So for studying this, we make or construct metabolic networks, and the KEGG is a very nice and well-maintained database for metabolic networks. And now there is this one Metasig. It is new, but here also people are updating the information about metabolites and their interactions. Next, we talk about signaling networks, like as I just talked. Signaling receptors or signaling networks, we talk about signaling molecules and the receptors. Now, here are also two different kind of things. Molecules can be proteins and molecules can be small compounds. Okay, like hormones, testosterone and other hormones are there, which are not proteinaceous in nature. And receptors are, as we all know, they are proteinaceous in nature. So there would be also this different kind of components are there in this network, and it is like a heterogeneous network. So here we are discussing about the edges are informing about the signal transduction. Okay, MAPK is there. It is interacting with some other enzyme for phosphorylation, PEP there. So what calcium molecule is binding, PEP is being activated, this like that. So in this kind of network, we talk about molecules, signaling molecules. They can be proteinaceous or non-proteinaceous in nature, and then there are receptors. So one receptor get activated, it activates another protein for another function, or another molecule that goes and binds to another protein for its function. So from extracellular surface, some signal is coming. It is being communicated in the nucleus, and in the nucleus, then there is some transcription factor which is activated that goes and binds to the gene. So you see how signaling network is connected with the gene regulatory network as well. Okay, so this is how beautifully a cell is working, and when we look at the network, we get a bigger picture. So these networks gives us information about signaling and cancer signaling and other normal functional signaling as well, and developmental pathways. Because in developmental biology, we study about signaling, that how the organ or tissues or organ development is happening, and it is being communicated between or among the cells of a tissue or between the cells of two different tissues. So this is that information comes through signaling pathways, and we study it through signaling networks. Now, in this example, this reactome is a very famous one for studying the signaling networks. And there, this signal link is also there, which is same as like Metasig. They are new, and they are also updating the newer information in this. Now, next is neuronal networks. This is also a type of biological network, and which is So we don't have to confuse this thing with neural networks. There is one thing called neural networks, which is in computer or AI and machine learning. So neuronal network is when we are talking about neurons, because as we know, in our brain there are billions of neurons are there and they are interacting. So each of them are interacting together. Okay, not each of them, but yeah, the neurons are interacting and forming a very close knitted network. So this kind of network is um, consist of neurons only. Now here we don't have anything else. Like two neurons are connected. Now two neurons can communicate through chemical signaling or through electrical signaling. Okay, it can talk, like one neuron can communicate with another one through chemical signaling. As we know, there are chemical messengers that they send one neuron talk to that another one. Or electrical signaling, we know that there are potassium and sodium gated channels are there, and there are calcium channels are also there. So the connection between two neuron can be a chemical or electrical. That depends, but the only component is neurons here. There is no other thing. So neurons, like, are there basically, there are different types of neurons. There are neurons and there are subsidiary cells also. So we can consider subsidiary cells as well, to make it heterogeneous and get more information. So, and also in this one I will talk about brain network as well. So by brain atlas, the brain has been divided into 92 different 92 or 93 different regions. Each region consists of many neurons. So there, here in neuronal network we are talking about neurons, but in the brain network we are talking about the region. Like one region is in my left hemisphere somewhere, and then another region there is left hemisphere frontal lobe and left hemisphere occipital lobe. So is there any information communicated between frontal lobe and occipital lobe? That is done through brain network. So brain network is different from neuronal network, which I have not mentioned here since we have brain network, neuronal network here. Now in this kind of network, we study the brain connectivity and information processing. That, like, there are many people who have go through seizures and epilepsy, those kind of things. So what kind of signaling is happening there? People study that how, why seizure is happening, or how the brain behaves during seizure and epileptic activities. So for that we use neuronal networks. And to get these kind of networks, you can go to human connectome project, and C. elegans neural map is also available. C. elegans is a very small worm. It's a nematode, and all the cells are mapped for C. elegans. It has uh, very few cells, and we have the complete neuronal map of C. elegans, and people study neural networks in C. elegans also. Next is we have a very commonly used co-expression network, which we get from the expression data, transcriptomics data. Like if you have the RNA expression values, we can convert it into a co-expression network. So here also nodes are genes, and edges are the correlation between the expression pattern of two genes. So with suppose one gene, we measure the expression pattern in suppose 10 different individuals or 10 different states. Okay, it's a time series. And second gene is also there. We calculate, measure the expression in 10 different states. So both the genes are giving some values. Now from this we can calculate the correlation between gene 1 and gene 2. It correlation can be positive or it can be negative. So negative means one gene is going up, other gene is going down, likewise. And if it is positive, both are either going up or both are going down together. So this is telling us that, okay, this particular function or this particular state, both the genes are going down. It means both of the expression of both the genes has gone down. One is going up, one is going down. Then it is like, okay, they are contradictory working together. Okay, so some gene is being activated, other is being deactivated, like that. So we look at the transcription coordination between the genes. and these are like highly studied and highly informative networks. People have been using this a lot nowadays, because of availability of RNA sequencing and easy access and cheaper price of RNA sequencing now. Yeah, people are making a lot of co-expression networks for different stages, different parameters, and making these networks. So example of these is WGCNA. Okay, one more thing, since correlation goes from -1 to +1, there are hundreds of values in between from -1 to +1. So this is having a weight. This edge is having a weight. It can be +1, it can be plus zero point 0.5, it can be 0, it can be minus zero point 0.7, it can be minus zero point 0.1. So, or it can be -1 also. So closer to 1 or plus -1 or +1 is strong correlation, and closer to 0 is weak correlation. So these are weighted gene co-expression networks are there, and this WGCNA is a library which is there, and these kind of co-expression networks are studied through WGCNA. Okay, now when we know that okay, this network is there, what are the things that network is going to tell me? Okay, we have suppose one big network there. What it is going to tell us? So now we have to enter into the mathematics of the network, like network properties. Okay, these network properties are being calculated through different formulas, and these are like very simple +2 level mathematics is there. No big integration differentiation is there. No calculus is there, because we are just looking at simple things here. So there are these are different properties which are at node level. Okay, node level means individual level, and there are some properties which are global for whole network. One thing is there. This is for each node we can tell. Okay, this degree centrality is for there for each node. Like I will explain right now. Degree centrality tells about neighbors of a node, which tells about how important a particular node, or for example in protein protein interaction network, a protein is based on its connections. So if you suppose search p53 on NCBI database, you will find suppose some 60 to 70,000 different entries, because it is a very important protein. Likewise, you search some other protein which is not that important, you will get some lesser hits. So here you can see degree centrality is there. You see the red node is connected with these six different people. So it means it is important. It is connected with these six because it is doing some very important function in the network. So degree centrality tells about essentiality of protein. Now consider a scenario. I remove this protein. These all individuals don't know how to talk to each other. This network will be disintegrated. No network will be there. All these six people will be free. So this particular individual in the center is talking to everybody and making a network and keeping the cell go on. Okay, so these kind of networks are seen as hub nodes. And in social media networks or such places, you will see that there are some people who have lot of followers and friends followers. If you remove this particular node, the network will get disintegrated. In similarly in biological systems, if you remove a hub node from the cell or knock out that gene, the cell will not survive. This is very direct information. Okay, next is the betweenness centrality. Now, betweenness means how important a protein is there to communicate between two systems. So, you can see here, this is one system, this is one system, and this particular individual is connecting these two systems, because it can interact with this node and it can interact with this node also. So this is giving us information about bottleneck and control point. Okay, this is a control point. For example, I know biology and I know data science, so I can talk to a core biologist, and I can talk to a core statistician also. So I am a link between these two. So these are called as weak links. So if you cut this, these two groups will not be able to communicate, and the and a communication will be stopped. Similarly, in biological networks also there are some proteins which have evolved in such a way that they are communicating between two different systems. Okay, so these like in signaling pathways they are there. Another one is closeness centrality. Now one is this thing that I am connected to many people. Then closeness centrality text talks about who is connected to highly connected node. Suppose I am friends with president of India. Okay, president of India has so many followers and all that, and I am also friends with him. Although I don't have many followers, but I am also a important person because I am connected to a important person. So that uh, characteristics is coming to me as well, like that. So in this kind of thing we talk about information spread. So somebody who is like friends with many people, if you tell that thing something to him or her, she can easily transfer the information to or spread the information to other people. This kind of networks, this kind of closeness centrality is used to identify metabolites in metabolic networks. Now there is another thing, eigenvector centrality. This is very purely mathematical thing, eigenvector centrality, and it is when we are talking about connectivity of neighbors also. Like, so this is suppose one person is there. This is the highly connected node, and this is a neighbor. So how these two neighbors are connected? So connectivity of neighbors also we look, and this is being used by Google's PageRank algorithm. Like when you search something on Google, it it gets some hits. It shows no 0.1 second to give 1 million hits. So it goes to all these things, and what to come on the top. I'm not talking about a sponsored one or that. Normally, if we it is not sponsored, what will come first? That is being decided by their eigenvector centrality in the network of web pages. And also this is used in human brain for fMRI data. fMRI means functional MRI. MRI, we all know what is MRI. F means functional MRI, when we look at the functional aspect of the brain and we do MRI there. So that is functional MRI. In this kind of data, we use eigenvector centrality to identify the regions, interesting regions. Okay, there is one more thing here we can talk, centrality and hubs. So centrality, so you see this is one, okay, this I will talk in the next slide. Okay, another other than that, node local or node level properties, we have global network properties, which are first one is density. So you see this network is very dense. Everybody knows everybody almost, like very closely knitted communities are dense. Here you don't get much information. The problem with dense network or completely connected network is we don't get much information here. Although these kind of networks are studied by physics people to study nonlinear relationships there. This is a sparse network, so less dense, and this kind of network is recommended in biological systems. So because we are only constructing the network, so we construct in such a way that we get a sparse network, because this is noise. Okay, this is noise. We don't want noise. We already are dealing with a huge data, and if we are also dealing with a noise, also we will not get information. So we want to remove noise. There are different algorithms or threshold methods we put to make the network from dense to sparse. And this information from this we study the network properties that we just discussed. Now, average degree is same as degree centrality. It is when we are taking the average of all the degrees of all the nodes. Like we have 10 nodes are there, and when we sum up the degree of all the nodes, it comes out of suppose 25, and we divide it by number of nodes. So 25 by 10 is 2.5. That is average degree. Okay, average degree is used to describe the sparseness. of the network, basically. Then there is the clustering coefficient. Okay, this is important because it talks about formation of triangles. Like me and Gopal sir are friends, and suppose Gopal sir is friends with someone, so there is a high probability that I will also be friends with that person. That is clustering coefficient. So this is like kind of a prediction that if two people are friend and one is friend with someone, there is high chances that this second person will also be friends with that third person. This is clustering coefficient. It is also simple mathematically calculated. No big mathematical equation is there. Next is average path length. Like from going from one node to another node, how many steps I have to take. Suppose I am going from this red node to this red node. This is the shortest path, means one, two, we count number of links, one, two, three, four, five. I need five, at least five links to reach that node. That is shortest path. So this is used for disease spread networks. When we construct the disease spreading networks, like in case of COVID, it is a communicable disease. One person coming in contact with the infected person, they can get infected. So these kind of this kind of information of shortest path is used in those kind of network. Second is when we want to construct power grid network. Okay, power grid is like electricity is generated at one power plant. From there, we have to take the electricity to different locations. So what is the shortest way I can reach from power plant to a city, to a far city somewhere? So there also this kind of information is very important, and people use this shortest path to construct those kind of networks. And then diameter is longest path in the network. What is the longest path? So we count all the from here, suppose this one or this one, we go here again. So we start one, then we go two here, three, four, five, 6, 7, 8, 9. So we are taking 9 steps. That will be like the diameter. What is the longest path present in the network? That is diameter. This is also the when only calculated for the whole network, not the individual nodes. Okay. So we here I will talk about now this centrality in hubs in here. There is another other than that those properties. There are structural patterns present in the network. So first thing is when we are constructing a network of proteins. Now we know there are suppose 1,000 proteins involved in a network, but some proteins are specific for some specific function. Under the broader picture of function, we have some specific function. So these forms a close clusters, and these are called as modules or communities, and it is calculated through modularity. There is algorithm for calculating the modularity also. These are also simple, very simple equations are there to calculate these communities and modularity. So high modularity means it tells about hierarchy also. Okay, hierarchy, like in the social system also we have hierarchy. So this modularity tells about hierarchy, that okay, one group of people are doing this thing, they are giving instruction to another group of people below them. More groups are there, below them there are more groups. So suppose some information is being given from the VC of your university. So VC or his some people are there along with him, some group is there. They decide on some new policy and they tell it to the HODs in the departments. So HODs will get that information. Now HOD will talk to the class teachers or class mentors, each class mentor, or there are program leads, they will talk to them. So in such a way, information is being propagated. So modularity tells about the hierarchy of the network, the arrangement of network in such a way that information is being produced at one point and being propagated at all the nodes like that. So this is also very interesting, and people in biological systems Our biologists look forward to these kind of patterns, that if there is any community formation happening, and this community, what kind of genes are coming, are they performing one specific function, or they are involved in one particular process in the cell. So these kind of things are very interesting to look. And next is assortativity. So assortativity means, this is assortativity gives up two different type of things: assortative kind of network and dissortative kind of network. So dissortative means high degree nodes connect with low degree nodes. So high degree, this is like high degree node and connected with low degree nodes. There are some nodes which are having like a high betweenness centrality is there, but mostly these are like high degree nodes connected to low degree nodes. So this is dissortative nature, this dissortative kind of network where high degree nodes are connecting with low degree nodes. Assortative network is high degree are connecting with high degree and low degree are connecting with low degree. This is a kind of pattern present in our society also, and in biological systems as well. That because some specific proteins or genes are involved in one particular function, but they are communicating with many other different proteins of that particular process. So this will give rise to modularity or community. Like this will form maybe one community, this will form maybe one community, and this will form two different communities. So this is how community, although this is very like specific, like only one is added, it is not like this in biological systems. It gives rise to modularity and hierarchy. In this you can see one is connected with all, so one can give information to all of them. It's a kind of hierarchy. This is very specifically they made it like that. But in biological systems you get dissortativity. uh, some sort of dissortativity is there, not very strong dissortative nature is there. Assortative networks are basically random, you can say. So this is also a global structural pattern present in the networks. Now this is next point is scale free nature of these networks. Biological systems are scale free. What is the meaning of scale free is they don't have any scaling. Scaling means you go from suppose you make a plot like this, number of degrees and frequency of that degree. So the nodes which are having low degree, like low degree, are high in number, and nodes with very high degree are very few. Like for example, social media network on Facebook or Twitter, there are some people which are having millions and thousands of followers. Okay, but there are so many people are there which have very few followers or connections, 100, 200, maybe 400,000. This is very high in number. So this is how scale free nature works. Like it can keep going on because you don't know. Suppose one person is having 1 million followers, next day it can have maybe 1 million 1,000 followers, next day it can have maybe 1 million 3,000 followers. So it will keep increasing here on this side. So there is no scaling here. Excuse me. And on this side also it will keep increasing because many new people are keep joining on the network, and they are having maybe 5, 10 networks, 5, 10 interactions. So this will here also it will keep increasing. On here also it will keep increasing. So there is no scale. So it is called scale free nature. And most of the biological systems are scale free in nature because new information is being added, because we are only doing it. We don't know completely uh, the all protein interaction networks or all gene regulated networks. We don't know it completely. So we are keep adding information there, and it gives a scale free nature to these networks. Now, networks are studied as complex systems. So system is anything can be a system. Okay, a mobile is a system, a watch is a system, a car is a system, a bike is a system. So that system is complete in itself individually. Okay, and it has many components in it. Okay, so engine, suppose in the bike there is an engine. Engine is one component which has many different components in it. So all those components are making engine work. Now, other than that, there is another thing is that like tire is there. So tire has many other components, but all together they are making one system. Similarly, we look at the system in terms of networks. Now I have got two different ways of signaling. One is you see a ligand is there binds to receptor. It gives information to one of this guy, and then this gives information here, and then this takes into the nucleus, and some downstream gene is activated. Very linear, one, and like that. Okay, you will get linear graph from this. But when you look here, you look at a complex picture of this network. Like one growth factor binds here, so many things are activated, some are deactivated, this is deactivating this. This guy is going somewhere activating something else, and here are other things which are being interacting together. So you see, this is a complex picture. Now I went to string and I got two different networks here. I typed in BRCA1 on the string database. It gave me one network and this is another network. Okay. So you see BRCA1 gene is here. It is interacting with this many. I am keeping it simple. Okay. We are not putting more information. So it gives a simple network. Okay. Not too much complexity here. Another network is like you see BRCA1 is here and so many proteins are there. And then this one is here which is one making one cluster. Okay, one cluster is there. So you see, we can have more information based on our requirement and availability of data. Since this data is available, we can get it. Otherwise, we don't. So a network can be studied through uh, complex systems. A complex system is studied through the network, and then we can study network in a simpler form or in a complex form, depending on our needs and availability of the data. So this is the real network which I got from String database. Like if you go and type BRCA, you will get this kind of network. There are multiple other options. Maybe we will have one workshop where I will talk about how to use String database and other databases which I mentioned today, and we will see okay this kind of network. So you will see there are two different nodes are there. So two different edges are there. One is pink, one is black in color. So pink is saying that these two are physically interacting. Okay, that experiment has been done. They found it. Second one, the black maybe is talking about their co-expression in the cell. So these are different ways. Like there is this one, it has three different edges. So we can define these connections in the string database. Okay. Um, uh, Gopal sir, we have time now.
यूजुअली 1 घंटे का करते हैं और
हाँ
कितने
सर,
टाइम लगेंगे?
हाँ मतलब I can finish any time because we started little late, so we have around
You can
15
take more
minutes.
10 minutes.
हाँ हाँ, so 15 minutes I will be done. Thank you.
ओके
Yes,
ओके
yes,
ओके,
sir.
फिर क्वेश्चन आंसर भी पूछोगे?
हाँ हाँ, so I will finish maybe in 10 minutes.
जी जी जी।
Okay, now there are different types of networks which are like undirected graphs or undirected networks are there. There is no direction between two nodes. You are my friend, I am your friend. That's it. Maybe in like professional setups, suppose I am working somewhere and somebody is my boss. So that is not undirected. Boss is giving me some information or work. I am doing that. So that is not directed. It is directed. So we call it like there is a direction between the bit direction of flow of information, like gene regulated networks. A particular transcription factor is either inhibiting or activating a gene. So that information is being directed. So that is called as directed graph. For all these type of graphs, same network properties we study. Although the some changes are there in calculation, but same network properties we study. Then there are unweighted graphs. Like in the second slide, I talked about binary matrix. Either it is link is present or not present. He is my friend or not my friend. Okay, that is unweighted graph. Usually, protein-protein interaction networks are unweighted in nature. Weighted graphs. Now, weighted graphs have the intensity of interaction. I am his friend. I am his best friend. So, best friend is there is intensity. Similarly, in co-expression network that when I was talking, the co-expression networks are weighted in nature. Whether two genes are having a strong positive correlation or a strong negative correlation or weak positive or negative correlation, that we talk through weight, and this weight gives more complexity for calculating the network properties. Actually, we have to tweak our all the formulas and algorithms to calculate that. Okay. Next is disconnected graphs. Now, suppose there are some node pairs which are there which are not connected to the major part. Okay, so there is possibility like you make one Facebook page, and only you and your friend are there. Nobody else is. You are not connected to anybody. So you have made one group which is not connected to the major chunk of the Facebook social media network. So this is called as disconnected graph. So when I was talking about here, sparse, making a network sparse. So we are removing basically some links, but we keep in mind that we keep the network connected. Okay, we don't go up to that limit that we remove some edges which make the network disconnected. Suppose this person is there, this node is there, or this protein is there, we remove this node, this connection, that protein will be gone. We will lose that from our network, and somehow we'll lose the information. So we try to keep the network as much as sparse that it doesn't make the network disconnected. Okay, so we this there are disconnected graphs also, and then there is complete graph. Everything is connected to everything. Okay, like five nodes are there, all five are connected to each other. That is not giving us any information. So in biological systems, we try to keep the network sparse but not disconnected, and we don't usually work with the complete graphs. Okay, like in neuronal networks, we get complete graph because neurons are very closely connected with each other, and they communicate. One neuron is communicating with many other neurons, and one like that. So there we get complete graphs, but there also we can do filter based on the uh, signaling. So these are different types of networks. Other than that, there are many other which I am not putting here, because then it will be little complicated to understand. Okay, now we can do some inference from biological networks. Like, okay, so this is very beautiful illustration which talks about how the genes are, how we can inference from biological. Suppose we have genetic interactions, so that also gives about the information of protein. Similarly, if you have protein sequences and we look at the protein sequence similarity and we make some network, that also talk about protein. And we talk about physical protein-protein interaction. That also gives us the protein information on protein. Okay. Even if we are talking about genetic interaction network. Finally, we talk about protein. Okay. These genes are being interacted and these genes are making this protein. So we are talking about protein here. Like protein genes are there. um, Other than that, we can have the information from samples, and this is basically the co-expression network. This is how it gives this kind of heat map we get. Like dark green is positive interaction, and white is negative interactions are there. So, or maybe we can just talk about positive interaction, and in one heat map, and in another heat map we can talk about only negative interactions. So 0 to +1 and 0 to -1. There we get the information of genes only. Okay, so this is like a simple representation that we just talked about. And another aspect is there when we are talking about gene regulatory networks. So this is basically expression patterns and all that. So that all, see here, directed network, we have arrow. So you see the transcription factor is giving this particular is activating this transcription factor, and this transcription factor is activating this gene. So you see square is gene, so this gene is being activated. But so you see, this arrow is very light, thin, and this arrow is very thick. So this particular transcription factor is strongly activating this gene, and this is like a little. I am also helping somehow, kind of thing. So this is, and this one is strongly activating this gene, but strongly inhibiting this one, you see, and this one is like strongly activating this. So this is directed and weighted, both are there. Now you can make all different combination, directed, unweighted, directed, weighted. So we can make all these things because this is completely in silico process, we are doing it by ourselves. So we can make all these changes. Here also it is undirected but weighted. You see, this is thicker and this is thin, so we can make undirected weighted also, and we can make directed unweighted also, depending on our filter. So this is just we talked, I'm just showing here this, like inference can be done. Also we can do some link prediction. So nowadays we know that machine learning has been booming because of availability of very good computational power and huge data. We are able to develop some deep learning or machine learning architecture using language models, and then we can do interaction probability prediction between the proteins, like you see here amino acid sequences there. So we can do perform the machine learning, use the machine learning algorithm to perform the prediction and get the prediction of links between two proteins. So we can do prediction, new link prediction based on the existing data. Without any existing data, we cannot work, because machine learning or anything works based on the data only. That's why people are building so many big, huge data centers and all that. So we look for the prominently used. These are like prominently used in association networks containing proteins, and then we use guilt by association principles are there. So guilt by association means, like, that's why I said no, like two are friends, maybe the third person who is friend with one could be friend with this also. This is very simple way of looking at it. So this is also used to for identifying the pattern of interactions. And then there are topological similarities also are used for link prediction, like we can do topological similarities are those properties which we just discussed, closeness centrality, degree centrality, eigenvector centrality. These are topological properties which are used. Now, other than that, there are higher order networks, but I don't think it is important here. So I will just skip this slide. Okay, and then there are some different network models are there. Okay, so basically three different network models are highly used to study the real world networks. So if you construct a network from your data and then you compare with the random network, with this small world network or scale free network. So this random network, or it is called as Erdos-Renyi ER random model network, which was proposed by Erdos and Renyi in 1949, and it is defined by probability. So connection between two is defined by probability. Suppose I set the probability of connection to zero point 0.6. So out of the whole network, 60% nodes will be connected. This is how, in simple way, we can say it is a very random, and it gives this kind of distribution. Some are having very low connection, most of them are having zero point 0.6 probability is there, that kind of connection, and very few will have high connection, high degree. Similarly, small world network is there. So small world network we see in biological systems, in brain networks, and small biological networks as well. There we see small world network. Small world network was says that the world is very small. If you are friends with somebody, and that is some with some friend, some with somebody, so you can be a friend with every person on this, every sixth person on this planet. Okay, this is how, in a simple way, we can say it is called as sixth degree of separation. So if you want to connect with any person on this planet, you can connect to them through your network after like six people. Like six people are there, in six is the path length for that. So low path length and high clustering is there. That gives us the small world network. And then a scale-free network, I just talked about this Barabasi, Reka Albert and Albert Barabasi. They gave this kind of network model, and it has no nodes and power law distribution is there. Now you see here, very few nodes have high degree, and most of the nodes have small degree. This is simple, and most of the network biological networks are like this. So these are the three different models which are being studied. And nowadays they are also using machine learning to do prediction of node labels, link labels, and subgraph level. Subgraph is basically communities in the modules in the network. And we apply supervised as well as unsupervised, both kinds of methods. If some of you are from machine learning, you might have known there are some supervised methods, and then there are unsupervised methods which we can apply on the network data itself. So it can be used for prediction of unlabeled nodes and links. Okay, now visualization of networks is there. So usually you see visualization is only done for giving a figure in the paper. Because looking at a network, suppose I show you this network, suppose you see this network, it is not telling you anything about properties, and no information is directly coming from it, although you can look at it, it looks beautiful and all that. So visualization is done for that process, but all the visualization tools have analysis option is also available with them. Like cytoscape is very commonly used, and now they have made it they have made it available with lot of plugins that you can apply, and you can get a lot of different information. So you can do visualization using cluster maker or GBay or mine plugins, and then you can do analysis using network analyzer and RIV analyzer and Centiscape for visualization for analysis. Similarly, Gephi is there, which is also written in Java and available for desktop, both Mac and Linux and Windows. And it is visualization and analysis both. So maybe someday I will do like this kind of also workshop where I can talk about Gephi and string cytoscape things, so that you know how to use all these different tools which are available. Then string DB is there, which is only on web, and you can study protein interaction networks. OmixNet 3D is there. So multi-layer, there are multi-layer networks also, which is maybe in some other workshop we can talk about this. So multi omics means genomics data, transcriptomics data, proteomics data, all different. These are there. So when you integrate all of them together, this is called omics. And when we integrate, we get multi layers. Like protein layer is there, and then gene layer is there. So gene and protein are also communicating, no? So that makes a multi layer network. That is also studied. Um, then Metascape is there for metabolomics. And other than these, you can make your own code in the Python or in R. So in Python, you use network X library, and it can be done for analysis and visualization both. Although visualization is not very good in network X, but analysis is nice. In iGraph library in R, the visualization is very nice, but analysis is not that good. So I use R for visualization part and network like Python network X for analysis. So that is up to you how you want to work, and how you want to create the figure, and how you want to get the information on network properties. Okay, this is a little bit about my work, that how I constructed the network. So I have used mitochondrial. I have worked on mitochondrial DNA sequences, human mitochondrial DNA sequences from like one of my work is this, that I used the three high altitude populations, Andes in America, Ethiopia in Africa, and Tibet in Asia. So these are high altitude populations from the three different continents, and I aligned the sequences, mitochondrial DNA sequences. Now you see, I am not using sequences directly as my network. I am using some numbers. So numbers is what? The position, mitochondrial DNA sequence position, whether 1, 2, 3. So I and J, any IJ pair I can make, and from this IJ pair, I have calculated a co-mutation frequency that also goes from 0 to 1, not minus, 0 to plus 1. Either there is no commutation occurring or it is strongly commutating. So you see like this TTT is there, which is major allele here in both cases, and CC is minor allele. But CC is occurring two times together, one time together. Here it is present two times, here it is present three times, but occurring only once. So in this formula, we feed this information 1 upon 3 into 2. So 1 upon 6. That gives us a commutation frequency. Suppose it is present, uh, suppose 3 times here and 3 times here. And all of them 3 times it is occurring together. So 3 into 3 upon 3 into 3. So that is 1. It means strong commutation is happening. Now you see, I am not using any protein or gene or nothing. I am using these numbers to construct the network. So that gives us a network of variable sites. Okay, position 1 is commutating with position 5. Position 5 is commutating with position 7. Likewise, and mitochondrial DNA is 16,000 base pairs, and variable sites you get maybe around 600 or 700, maybe you get 500, maybe you get 200 sometimes. So 2 out of 200 you can make a network of 200 variable sites. Now these variable sites can be used to get the information of genes, like because each variable site or each this ATGC is also a part of some gene in the mitochondrial DNA, so we get the information of gene. So from here, this is just and this yeah, this is just the network properties which we discussed. Like number of nodes are there. So 398, 837, 1197, like that. You get average degree 17, 33, 34. So out of 398, on an average, each one is connected to only 17, and there are 398 nodes. So it is very sparse in nature. Okay. Average clustering is high, so zero point 0.8, zero point 0.7, zero point 0.8. High clustering coefficient is there, which is good for forming the communities. Modularity is zero point 0.7, 0.5, zero point 0.4. So Tibet has high modularity. And I wrote all like what is the biological inference of all this. Then average path length is like this, and degree correlation, which is associativity. So this is only negative, like negative associativity is there, so more hierarchy, and this has positive is there in this one, in these two. So these are like simple network properties we calculate. But being a biologist, we have to write the biology of this. Okay, why? What is the meaning of zero point 0.7 modularity? Why it is happening there? What kind of migration or what kind of evolution has ever happened on Tibet? What is the adaptation pattern? We all write this thing based on these numbers only. So yeah, now from this there we make the gene networks based on the variable sites. Like ND5 has suppose some variable sites belonging to ND5, and those variable sites are interacting with those variable sites which are belonging to cytochrome B. So we make ND5 is connected to cytochrome B, and bigger the size of this node, it means it is having high degree. It is important in the degree centrality is high, like ATP6, ND1, ND2, high degree centrality. Here CO3, ATP6, ND3, high degree centrality. Likewise, similarly, we get the these networks are from exclusive nodes, which are only present in Tibet, not in Ethiopia or Andes. This one are from only from Ethiopia, not in Tibet and Andes, like this, exclusive to this particular region. These were from common variable sites present in this, and this, like that. These one are exclusive to that particular region. Now Tibet up and down means these are these interactions are uh, having more weight than the random. I discussed random network just now. So we construct the random network, then we compare, and we see okay, this weight is higher than the random, this weight is lower than the random. So this is lower than the random one, this is higher than the random one, because we don't want the random connections here. We want
अब
something.
कितने स्लाइड्स हैं?
Yes, sir. Real last step. This is just
Good.
last slide. So like this we get, and then from this we do functional enrichment analysis. That okay, this genes are I found important. What is their function in Go gene ontology terms or KEG pathway? We look for these genes, and we get some information from there, and we write our analysis. So this is how like networks are constructed, analyzed, and studied, and finally we give some inference from them. And with this slide, I finish my presentation. I think, yeah, I took little 5 minutes more, but
कोई बात नहीं, कोई
thank
बात
you
नहीं,
for
अच्छा
this.
है।